I heard is actually quite consistent what I think Linda Fried and others pointed out, what they call dysregulation of systems. This is homeostasis, which is regulation, right? But you still haven't given me the reason why you would want to put a price. And after all, why not put out grants or work with the colleagues here or with NIH and to say, this is an area we need more research. Why a prize?
Why prize? Well, prizes you get to set a novel target. It's a little like canoes. When you push a canoe, which is like grant making, it can go in all sorts of different places, some of them very wonderful. But it can often miss the target. When you pull a canoe, it comes straight at you. So prizes are a mechanism to pull innovation. It's like pulling a canoe, so it comes right at the target you're intending to do. And prizes started, I mean, there's been many prizes over the years, but the Valley has actually been one of the leaders in prize formation. This is one of the things I think we should recognize. The Valley's had a huge role and interest in this field, in part because of the technical innovation and the entrepreneurship.
Right.
But also, we're also in a time where there's tremendous sharing of information that's going on that's enabling new ideas to emerge.
Right. So one thing that I think all the panelists have in common is that they weren't working in the aging area, right? I mean, you weren't working in the aging area before, where there's in fact some of the speakers today have spent their whole life working on it, and yet all of you came into this area recently with a belief and a bet that in fact something good can happen and great can happen with you. So Craig, as I think about what we talk about today, you know, when you think about aging, it's not as if it is a unidimensional activity that, you know, you go from here to there. In fact, when I think about I think it was your when I talked to you, a pole vaulter who has cognitive problems, or a sharp-minded individual who requires a wheelchair. So the biology is very different, right, overall in terms of when people age, and particularly which organs, which tissues, what areas of infirmities. So do you think your technology with that information can actually predict the differences between individuals, and not only about the rate of aging, but in fact the various diseases and organ degeneration that people see in a variable rate and difference between individuals?
Well, we start out being all totally unique. We can even at the sequence level tell identical twins apart just due to the somatic variation that occurs. So everybody was looking for magic bullets of the single genes that you could turn on or off that would affect biology. And biology doesn't work that way. It's changes across the entire genome. Your variants are totally different than mine. We all start with a different set. So you're right, no two people are alike and they don't have the same conditions. If your mismatched DNA repair enzymes are mutated, your chance of getting cancer go up, and you have other problems with homeostasis. Our genetic code degenerates faster. But it all gets back to our cellular programming and the changes that we see in the genome. If we could find a way to prevent that from changing, we might change all the processes that you were just describing. But it's a coding issue. But we can use the information intelligently in a preventative health fashion. And I've had my own life saved by having a prediction of a high risk for melanoma. I'm in the 93rd percentile in my genome. And that doesn't include behavior like paddleboarding and surfing and sailing, which probably makes it a 200 percentile category. 10 years ago, I discovered a small melanoma on my back. It was surgically removed very simply, and that was the end of it. Had I put it off for 6 months or so, it would have cost my insurance company a few $100,000, and we probably wouldn't be having this conversation today. So information is power. If you have information about your operating system, what's unique and what's different about it, you can use that in your life if you have that information.
Yeah, so to get sufficient information, as you point out, you got 20,000 sequences, genome sequences, but the phenotype is the big issue, right? And you've described some very detailed phenotype, like your T3, the magnet, et cetera. How do you actually get enough information about aging? You know, trying to collect all that information, and how big is the data set that you need to actually enable you to say, and I know it's not only genomics, you're doing metabolomics and all this stuff to say, yeah, I can dial that in information on individual, and I can really get that prediction about that whole next 25, 30, 40 years of the condition of the individual.
So one of the biggest surprises is when we pass 10,000 genomes, common variants in the database saturated. So everything we're discovering now are extremely rare variants in the population. But it's the rare variants that make us totally unique. The ALK translocation that determines whether the Pfizer drug will reduce your lung cancer is about 1 in 400,000 in the population. It's about 5% of lung cancer patients. So it's these very rare things that are going to give us the unique regulation, unique phenotypes. And I've always assumed getting to 1 million genomes was sort of the starting point, not an end point. I've been absolutely blown away by how much machine learning can discover with a much smaller database. So the discoveries are happening a whole lot faster once we pass 10,000 genomes.
Hal, you introduced me to a naked mole rat. As I said, we're going to make this the mascot of this meeting. But you know, earlier Cynthia Kenyon did roundworms. And so how good are these model organisms? Someone says maybe the best model organism is the human being, right? So talk to me a little bit about the research approach that you're going to take in looking at all this various, as you said, comparative genomics or comparative biology.
Yeah, I think it's a great question. People often ask, you know, should you study animal models or the human? And I find that fundamentally a false choice. There's no doubt that you have to study both. But the typical bench to bedside paradigm, or the paradigm that says it's the bedside only, I think is wrong. What you can learn from an animal model, you can then assess whether that abnormality or that finding is in fact observed in humans. Or you can find something you observe in humans and then ask, is it epiphenomena? Is the high levels of factor X that's associated with disease causing the disease or actually protecting you from getting worse? And so you can have human observations that you can then learn from the animal models, whether they be mice or naked mole rats or worms or yeast. And it's really an iterative cycle of bench to bedside, bedside to bench. And that's, if you look in drug development and advances that have been made in science, it's that iteration that I think...
You're collecting as much information as you can, and then you find out whether they're relevant or not.
Yeah, you're learning from all the models.
Right. Joe, I think it's very clear to me that the approach to this has to be multidisciplinary. Talk to me a little bit your vision about how to bring all this biology into your digital technology and your remote, you know, all that stuff, robotics, you name it. Yeah. I mean, what's your vision about bringing everything together? Because it's been said this morning, things are somewhat siloed, right? People are doing this pathway, but I believe that actually the opportunity is to bring all this together.
Yeah, I mean, there's a fine line between vision and hallucination, so I might border you know, I think we've been making this false discussion about, you know, whether we need more researchers in STEM fields versus non-STEM fields. Multidisciplinary approach to a multidisciplinary sport, that is aging, is an imperative. We need to have the biologists talking frankly to the social scientists, because I can find out from the folks up here whether I chose my parents badly or not, but I need to have some insight in terms of behavior so that I can find that melanoma ahead of time and take action to do that. I think the technology that is coming along now in your home, in your car, on your wrist, and soon inside you will give you the information to notify you that, you know, you haven't been eating well, or you may be dehydrated, or you take your medication and the like. But that multidisciplinary perspective is not so far going to help us with a big problem, which we've been talking about on a scientific level, which is information that we're putting into the hands of the individual and the family. It's too much. And so I think we're going to need to come up with creative ways to put bits and bytes of information that are digestible for all of us. And that's going to take English majors, biologists, physicists, and indeed engineers.
Well, so where's the place that would fund research like that, right? If I write a research grant, I go to NIH, yeah. And you may go to NSF or whatever, and we all go different directions. So that leads June to this whole conversation. I mean, everybody's in agreement that this should be multi-sector, right? But now, you know, Richard Holders tells me that the NIH, his institute's funding is $1.2 billion a year out of what, 30-some billion dollars of NIH funding? And if you look at total funding, so how do we think about bringing the right resources together?
Well, one of the things that would be really useful is to make the area investable. And for private sector investors, thinking about longevity, you got regulatory issues, you got public perception issues, you got biomarker issues. So one of the things I would love to see is to develop more proximate targets, such as development of biomarkers, development of interventions that improve those biomarkers, so that we can backdoor into longevity, but improve something today, where the benchmarks are achievable today, assayable today. So we're essentially focusing on health. If we're focusing on homeostatic capacity, we're improving health. Most people are for health. And then if we get a longevity as a result, as a backdoor win, that might be one way to make this area investable, draw a lot more venture capital into it, a lot more philanthropists who want to see near-term results, because it's very hard to show long-term longevity in a human lifespan.
We are open for questions, so please go to the microphone. And while you think about your questions, I do want to ask Hal. He said about moonshot. Okay, help me there. How do we get the moonshot off the ground?
Well, I think this is a good start, this conference, and bringing up the issues. I think what June said is really important. One of the things that you need to have happen to get a moonshot really get traction is to have some quick wins as well. And it would be really ideal that maybe from this meeting or subsequent to this, that people take the idea of biomarkers and being able to predict a person's biologic age. I mean, everybody knows their chronologic age, hopefully, but virtually no one knows their biologic age. In fact, we probably would all argue that we aren't aging at the same rate. But if we were able to create a way so that we could all, kind of like cholesterol is for vascular age, if you will, your biologic age, then people could imagine that interventions could be assessed in a much more timely manner. That might make this field seem much more tractable. It might identify areas of biology to go more aggressively into, and that might spur both the private sector as well as potentially government to invest more in these moonshots.
Yeah, I'd like to come back to that question, but we've got Dr. Druckmann first.
So I have a question for Craig Venter. I heard you mention just barely something about identical twins. I am an identical twin, and I'm wondering how many of your 20,000 genomes do you have of identical twins? And do you plan, in fact, to look at that?
No, in fact, we've been sequencing from a large cohort out of England where they've been following the twins for over 20 years. So we got all the phenotype data with them and in exchange for doing their genomes. The problem is the standard of measurements of phenotype data 20 years ago versus today are very different. And so while we have digitized health records now, all it means is you don't have to read the doctor's handwriting. That's about the only difference.
And you get much less information.
Yeah, but twins are an essential part of medicine and particularly genomics. We're trying to see if we can tell the difference in twins predicting your photograph and voice. So we're not so sure about that one yet.
Thank you. Questions?
Alexa McRae, Harvard Medical School. I'd like to pose a question to at least 3 of the 4 panelists, which is that you've talked about innovation, you've talked about collaboration, you've talked about the difficulty of collecting data, but the importance of having very large-scale data sets in order to take the next step in innovation. And what I'd like to ask is what commitment are you all making to making those data openly available so that more and more people, not just the elite whom you have mentioned in your organizations, can have access and can help us collectively move everything forward?
Go ahead, Craig.
So it's an issue we're wrestling with quite a bit. So if we sequence your genome, we have your genetic code. If we have your clinical records, we have that. And we have a lot of phenotype data on you. In theory, you can de-identify that data by not having your photograph with it or your name or address. But if we can predict your photograph and your voice and a lot of other things about you from your genetic code, who do you want to have access to that data? Do you want it to be on the internet and let anybody track things about you? Right now, HIPAA regulations say you can de-identify a genome as long as there's no photograph with it. So having open databases that anybody can look at is a real fallacy when it comes to medical records and precise information about each individual.
And I do understand that argument. The question, though, is if I am willing to make my information available on the assumption that there will be responsible use of those data, then we ought to allow those data to be made available in large scale.
Yeah. My genome's been on the internet for 15 years. Some people are making responsible uses out of it, others are not. So you can't, once it's out there, you can't control it.
I want to predict a few things about you. Joe, you want to answer the question? Or?
Yeah, I mean, we make, I'm sorry. We make our data available both not just through publications and theses, but for the data sets we have, for instance, on the physiology of older drivers and the like, that's available either on our website or just tapping one of our researchers.
I should tell you that because this is an area that we're very interested in, National Academy. We had a report on sharing of clinical trials data. Certainly, as you know, yes, and as you know, there's also GHG4 or G4GH activities. So we're going to keep discussing this, which obviously is a critically important issue for us and for society.
Thank you very much.
Question.
Yes, it seems to me that your marvelous research in immunology and Alzheimer's disease can only inevitably lead to an increase in longevity, at least for the 1% of Americans who can afford what I presume will be the astronomical cost of your therapy. Have you ever wondered if an increasing population, which I think will result, may be the end of humanity as we now know it, perhaps calling it the law of unintended consequences?
Who'd like to take that?
Well, so interestingly, one of the most interesting demographic trends to emerge from the longevity movement is that when people forecast long lives, among the other things they remodel and stretch out is actually their fecundity. So when you expect a 100-year lifespan, do you really want to get married when you're 20? I mean, do I really want to be with this person for the next 80 years? No, I want to do more due diligence. So essentially, people delay their life history strategies. And so right now, 100% of the high longevity countries are actually experiencing sub-replacement fertility rate. So if anything, we're seeing stabilization. Now, that doesn't mean it's going to continue in the future, but so far, so good.
By the way, the answer to your question is the highest divorce rate is amongst the 50 plus. So the answer is no, they don't want to hear us for 80 years.
Nancy.
Nancy Adler, UCSF. And the last session we heard about the marked disparities in aging by socioeconomic status and race and ethnicity. Of the things that you're working on, what are the things do you think might lessen that gap? And what are the things that might actually exacerbate the disparities that exist? And if so, what might you do to avert that?
Al, you want to take that on?
Well, I think, excuse me, I think the first part of this, and I felt this way being a novice to the aging field, was that one needs to really understand, I think, not just the socioeconomic predictor, but what's the underlying biology? Is this something solved by greater education, in which case that could be applied, or is there some biology that's going on that's a result of a lower socioeconomic? Is it food intake? Is it this, that, and the other? And so I think that there's an opportunity, depending on what you really learn by going deep into the biology, that gap could be decreased. I guess it's theoretically possible that when you understand the biology, that at least interventions that are learned from this biology don't affect that gap. But I think what I've taken away from much of today and my sense of the field is we really have a lot of unanswered questions as to why certain things are associated with certain things. And really getting at the heart of that, at least for me working at Calico, is to really understand the biology behind why is that discrepancy there.
Thank you. Anyone else want to answer? Yeah, great.
So the disparity comes in part, we have drugs now for treating cystic fibrosis that cost over $300,000 a year per child. That creates an economic disparity. And the same chemicals, the same test can be available across the economic spectrum. So we worked out with Discovery Health, a South African insurance company, because of the income differences, a program where they start to get their whole exome sequenced for $250. Doesn't have to cost $5,000 or $10,000. So we're trying to work on both ends of the scale. I think the pharmaceutical industry, working with the insurance industries, have to make sure both ends of the spectrum are equally covered.
And I guess the
Thank
other
you.
thing I'd
Yep.
say, with all the smart technologies that are coming into the home, the car and the like, just think of how much you paid for a computer in the 1980s versus today. Eventually those prices do tumble. That's the natural curve of technology, and hopefully that'll improve the equity of access to the quality of life that these things can provide.
Linda.
Hi, Linda Fried. I'd like to ask a question that is somewhere at the intersection of cathedrals, moonshots, and canoes. I think that the need for public-private partnerships in science is profound, and the opportunity is immense. And you all are in different ways investing in an understanding of taking the long view on science, even if you're trying to find some short-term wins in that. Because great innovation depends on being able to do that, as you well described. I'm interested, given the entire scope of the day today, in what you think is the role of private investment in science, of public-private investment, and public investment. And all of the principles that I've heard laid out about the science of the future, needing to be multidisciplinary, needing to invest in social science as well as basic and population science, working together, is not what I see going on in the public sector investment in science. In fact, it's a narrowing and narrowing and a shortening and shortening of the time frames for science. So if there is a role for public investment, such as through the National Institutes of Health, what do you think are the kinds of questions that publicly funded science needs to be asking and answering? And what's the difference in the kinds of questions we should be asking through public-private ventures or privately funded moonshots?
You want to take that on?
Well, I can start anyway. So between the public and private domains and institutions, there's a different risk tolerance. And you can argue in the public side, they could be taking more risk given the purview they have, but in fact we know that academics tend to be relatively risk averse. So where the private sector can come in is fill in different parts of their risk spectrum. You know, if you said a few years ago that aging was a disease, it was hard to get funding around that narrative, whereas on the private side, you can take more risk on things like that because there's a gap. So wherever you see gaps, the private sector can fill in.
All right. Yeah.
Let me just add, I think it's such an incredibly important question, and one that if there was an obvious answer to would be phenomenal. But I think a lot more work has to be spent thinking about the answer. My own perspective is that what's missing in my experience is that there's fundamentally a true lack of collaboration, even when people are working together. Collaborators typically deeply respect each other for what they bring to the table and contribute in ways that maximizes what both are trying to get at. And I think if we can really develop those true collaborations between the public and the private, and everybody really focuses, using their own models and their own drivers, on trying to solve this aging problem, 20 plus years from now, we'll make a good progress. And I think there's examples where we have these relatively large longitudinal databases that the NIH in their wisdom have created and sponsored and helped put together. Those are very, very valuable assets that maybe there's not resources with the public sector to make those optimized, to learn. But those are places where collaborations might be engendered to produce results that neither alone could ever imagine happening. And I think that's a place to start.
Yep, I think we need to figure that one out, because I do think that's a really important part of the solution, is how do we make it work? And presumably this will be another conversation. But let's have the last question. Last question from Deepak.
Deepak Srivastava, Gladstone and UCSF. I imagine the machine learning approach you described for looking at the genomes involves a lot of pattern recognition to predict the features. And I wonder if you can comment on what advances have to be made to go from that information to the genes and biology information that somebody like Hal would need to make new therapeutics and new targets.
No, it's a great question. So machine learning can find associations between different events or different data sets that you might never find through intuition or any other direct action. But it actually finds things in the genome the way our genomes work with biology. It's information across the entire genome. It doesn't look for an individual SNP that tries to explain everything. So it's not so trivial to deconvolute the machine learning data into a diagnostic assay of measuring something in DNA directly. So, but it gives you the associations to start with that when you hear about them, they make sense. I wouldn't have started, for example, looking for links between the shape and the measurements of the face and your voice. When you hear about it, it's not illogical, but deconvoluting that to the exact parts of the genome that code for your face and how that ends up influencing your voice is not part of the linear world that most academia exists in right now.
Thank you, and I want to thank the panelists for an outstanding session. John. So this actually concludes the program, but I do want to thank the program planning committee, Al, and your whole team. Thank you. The membership office for putting together this outstanding meeting and the activities of the last few days. Of course, my personal staff, my chief of staff over there, Morgan Kenrack, and of course the Office of Communications from both IOM and NAM, and many, many staff who you saw volunteer the time to make this work. So please join me in thanking all of them. So we have a reception, which is right in the Great Hall in the West Court. Please join me in having a drink and relax.